So I would take a very simple and well-defined question and would examine how you could approach this question using these modern techniques. And the major aim is to keep in mind that all these modern techniques are really excellent and great tools, but as all the new tools have many pitfalls. And all along the lecture, I would like to draw your attention to these pitfalls. So whenever you get into or start these experiments, you must bear in mind what you should control. So in the first slide, I would try to start with this bold statement, which is now obvious to everyone. And actually, really, students of this decade must bear in mind that we are really undergoing a major revolution in neuroscience. I really wonder if 20 years from now we look back, how we will regard this era. I really remember my PhD time, and compared to this era, it was basically stagnant and nothing happened. So basically every week you discover something very jaw-dropping new approach, and the combination of these approaches, it's really amazing. So as we all know, you can use optogenetics for activating well-defined neuronal elements, and as we also saw in the previous talks, it has a very high spatial temporal cell type specificity. This is a typical example of how you can activate a given cell population if you are good enough in an all-optical manner. Let's talk about another new technique, the pharmocogenetics, where basically it's possible to modify a single molecule in a spatially well-defined cell population. An example of this technique would be, which I will also show, that you can focally knock in or knock out a single gene in a given cell population in adults, and as a consequence you can avoid all the pitfalls of the full knockout animal. Of course, this technique doesn't have the temporal resolution as this one, since to remove a protein from a cell population requires at least a week. So the questions we will tackle is linked to sleep spindles. Sleep spindles are one of the most studied cell assembly activity in the brain. The most important feature is that it is transient. So if you look at a human EEG, when you start to fall asleep, exactly when you start to lose consciousness and the connection with your then transient 5 to 12 or 15 hertz oscillation emerges, and they last around 1 second or so. But the very interesting feature is that the duration is highly variable. So a spindle may last only for 4 cycles or up to 20 cycles. And apparently, this duration is a very important code, since if you learn something during your awake period, the successful recollection of these data is significantly affected by the duration of the spindles the following night. So the longer the spindles, the better you will remember the next day. So apparently, it has a very important role in post-learning stabilization of the memory trace. The sleep spindles also change a lot during ontogeny. Young adults have much longer spindles, and as we get older, spindle gets shorter. I'm not saying whether it has anything to do with the learning ability, but this is the fact. And most importantly, the duration of spindles is changing in lots of different neurological diseases, including schizophrenia, mental retardation, and so on. So the question which we will ask in this talk, what determines the duration of these leaf spindles? What are the molecular and network mechanisms basically tells an oscillation to stop? And of course, you may ask that in general, how the length of any transient oscillation in the brain is determined. If you think in the brain, almost every oscillation is transient, be it gamma, sharp wave, ripple. It starts and stops somewhere. And we know many things about how it is emerging, how it is maintained via the interaction of neurons, but we know very little how this oscillation stops. So sleep spindles are generated in the thalamocortical circuits. Just to make it very brief, it consists of excitatory thalamocortical cells, the thalamic cells which project to the cortex, also excitatory corticothalamic cells, and there is a major player here, the reticular thalamic nucleus. These are inhibitory cells which are innervated both by thalamocortical and corticothalamic cells. So very early studies have already showed that the spindles is basically arise as an interaction between the excitatory thalamocortical cells and the inhibitory reticular thalamic cells. So if you simply lesion cortex, spindles still go on because these two neuronal populations can talk to each other. So how it goes? It was shown as early by 1997 that there is a very unique ping-pong mechanism between these two cell populations. So as we fall asleep, the TC cells, the thalamocortical cells, hyperpolarize, and they get into a bursting mode. And when they burst, they will activate RT cells and NRT cells, and NRT cells will also produce a burst. This burst is very, very strong and inhibits TC cells and prevents their firing for all of time. However, TC cells have very specific rebound mechanisms, and after this inhibition, they again produce a burst and the whole cycle starts again. So the basic idea that excitation, inhibition, with this ping-pong mechanism, the oscillation goes on, and basically the time course, the 7 to 15 hertz, is determined by the kinetics of the NRT-mediated inhibition and the rebound. Of course, the big question is that why the whole thing stops after 4 to 20 cycles? Why rebound stops after a while, or why the inhibition stops after a while? And this is what we are going to study in this case. So the experimental strategy, which was developed by Peter Bartow, is to insert silicon probe to the among the thalamocortical cells. As you see, the fortunate situation that the reticular nucleus is basically spatially segregated from the thalamic cells. So if you put an electrode here, of course, you may record the somatic action potentials of thalamocortical cells. But what Peter discovered is that by spike sorting, you can also record the axonal potential, the axonal action potentials of the inhibitory reticular thalamic cells which innervate these cells. So basically, with the same electrodes, you can record the activity of the interconnected excitatory and inhibitory circuit. I don't have right now the time to show all the proofs that we were really recording axonal action potentials, but all those who are interested, I will tell at the end of the talk or maybe in the... So this multi-site silicon probe is a very useful tool for these recordings. And these are the sorts of results we will get. Here is a spindle. We will sort them according to the cycle number, basically according to their duration. And at the same time, you will record both TC cells and NRT cells, and you can examine the cycle-by-cycle dynamics of their activity throughout the spindle. And as the first look, indeed, the first data quickly confirmed that the ping-pong mechanism is true and valid. So what we did, we examined in which phase of the spindles the individual cell populations fire, and this is a phase polar plot. The peak of the oscillation, a peak of a spindle cycle would be 0 degrees, and anything which comes before this is towards this direction, and the other one which comes after is this direction. So basically what you see that in case of every cycle, first you have a TC activity followed by NRT activity, and then a long silence. And this is translated to the phase histogram that first you have the black is the thalamocortical cells, they excite the inhibitory cells, that's why they come later, and then a long silence and the rebound. So apparently these data clearly demonstrate that you have a ping-pong mechanism, excitation, inhibition, rebound. So far so good. And as you can see, indeed, inhibition always follows excitation in a cycle. Is it clear so far? Excitation, inhibition, oscillation, interrupt. So now what we want to ask is that how these NRT bursts actually interfere with the spindles. What is the exact role? And for this, basically, we have to interrogate the GABA-A receptors. As this is a very tricky business, since, as I will show you later, there are no specific antagonists to do this. So what we first plotted, how the NRT bursts changes as the spindle progresses. So we selected spindles with 5, 6, 7, 8 ETS duration and examined cycle by cycle how the spikes number within individual bursts changes as the spindle progresses. And of course, the data clearly show that in case of all spindles, the spike of individual bursts contains lots of spikes, and the number of spikes is diminishing during a burst. So basically, in the beginning of a spindle, you always have a lot of spikes in an NRT burst, and in the end, you have very few spikes. In case of the TC cells, you don't see any big changes. So the big question is that what happens with the GABAergic inhibition as this spike per burst decreases during the spindle? To investigate this, we have to look a little bit at the molecular composition of the GABA-A receptors. As many of you probably know, it has a pentameric organization and contains many, many different types of subunits, but it makes it almost hopeless to study them, since you could have thousands of combinations. But hopefully it has been shown that there are two major GABA-A receptor types in the brain, so not all combinations exist. One of the most important ones is the synaptic GABA-A receptors, which always contain gamma-2 subunit. And they are present in the synapses. These are the classical synaptic receptors. They are activated with very fast kinetic link to an action potential. The little less known receptors are located extrasynaptically. They contain delta subunit and never gamma subunit. And as you see, they are outside the synaptic cleft. And this, of course, causes lots of difference between them. The synaptic has very fast kinetics. They are activated by the presynaptic activity. And as a consequence, of course, they have major roles in timing and oscillation. In contrast, extrasynaptic receptors basically have a very slow kinetics. It's a tonic current. It's almost like a potassium leak current. And they are activated not by the synaptically released GABA, but by the ambient GABA level of the extracellular space. and they have a major role in regulating the excitability in a tonic manner. So that's why it's called tonic receptors, whereas these guys are called phasic receptors. Of course, we should study them separately, but as I told you before, there is no specific antagonist of synaptic versus extrasynaptic receptors, so you cannot study their role in EPSC generation. If you do simple gamma-2 KO animal, then it's lethal since it has so strong epileptic activity that it dies, I think, within 1 day after birth. So our approach was to eliminate gamma-2 receptors only from the thalamus and examine what would be its effect. So we use this viral-mediated focal knockdown of synaptic GABA-A receptors, which contains gamma-2 subunit, using the Loxp-Cre recombinase system. As you probably all know, for this you have to generate an animal in which the desired gene is fluxed between two Loxp sites, and then you have to inject a viral particle which contains Cre, and in this case, if we have a region-specific knockout, so the synaptic GABA-A receptors are basically missing only from a very well-defined portion of the thalamus. For this, you have to wait 8 to 10 days, since the protein synthesis is blocked, of course, since you don't have the gene and the proteins are eliminated. Of course, the first thing you have to do is the proper control whether the intervention is really working and whether it's really selective. Can we switch off this lamp maybe? Because I think it's, can you switch off this lamp? The first one. So what we do here is we inject Cre virus and as a control only GFP virus and examine the expression of the respective subunits, and hopefully you still don't see quite clearly, but you expect to see very high level of gamma-2 receptor expression here and basically none in this case. In case of the alpha-1 receptors, you see high expression here and a strong reduction in the knockout, and this is basically because alpha-1 subunit is linked to gamma-2 subunit. So if you remove gamma-2, there will be, thanks, it's better, there will be a reduction in the alpha-1 subunit as well. But finally, and most importantly, alpha-4 subunit labels the non-synaptic receptors, and the level of alpha subunit has not changed. And this is the quantification: major drop in gamma-2, significant drop in alpha-1, and no significant change in alpha-4. So far so good. We have subunit selectivity and specificity. Of course, we have to go one at a higher magnification because maybe the remaining gamma-2 subunits are still accumulated in the synapses. And to check for it, we use the super resolution storm microscopy. In this case, of course, the scale is only 200 nanometers. So this is one GABAergic terminal labeled by the vesicular glutamate transporter. And the red spots are gamma-2 immunoreactivity and basically indicate the accumulation of gamma-2 receptors in the synapses. So these are synaptic staining. And as you can see, there is an axon terminal establishing a single synapse here, whereas this GABAergic terminal, NRT terminal, established two synapses. In the Cre animals, Cre-injected animals, of course, we see the terminals, but we never see gamma-2 positive clusters around the terminals, indicated that indeed all gamma-2 subunits has been removed from the synapses. You have to do, however, functional identification of the effects, and to do this, you have to measure whether indeed all the phasic iPSCs are gone following the pharmocogenetic manipulations. And this is important since you still have to wait 8 or 10 days, and there may be some compensatory mechanism, and maybe other gamma subunits may be upregulated. But fortunately, in our case, it was clear that this is the control case. Gabazin blocks all spontaneous iPSCs. These are the Cree-injected animals, and as you can see, basically there is no downward deflections. There are no GABA-E iPSCs in this preparation, indicated that indeed we were able to eliminate all fast inhibitory action, and we confirmed it also by evoked iPSCs, so no evoked iPSCs in the Cree. So apparently, we were able to do an animal where focally we eliminated all conventional phasic inhibition. And of course, now comes the exciting question. These were just techniques so far. So what happens to sleep spindles? Basically, we eliminated a major portion, we thought, a major portion of the circuit, the synaptic GABA-A receptors. So the experiment goes on like this. These are the control experiments. In this case, we use a linear silicon probe to be able to measure the generation of spindles in a larger tissue volume. So these are individual recording sites. And as you can see, in the filtered multi-unit activity, from time to time, there are transient oscillations, which are basically the sleep spindles. In the bottom panel, I show you a wavelet analysis, which is a kind of spectrogram, which shows that during the spindles, the spectral density of this oscillation is very high, around the 10-12 hertz range. So indeed, this is how normal spindling animals look like. And you see spindles are quite coherent among the recording sites. And now comes the big advantage of the focal injections. As you can see, we can really penetrate the virus injection sites with our electrodes. And we can be absolutely sure now that this patch of tissue does not contain synaptic GABA-A receptors. So what happens to the spindles? Do you see it? No, you don't, because basically nothing. So this was one of the most shocking data I ever had, because we wanted to try these experiments under conditions which we were basically sure that it should work. I mean, if oscillation depends on phasic GABAergic inhibition, you must understand that I'm coming from a Hungarian tradition of long history of studying interneurons and oscillations. And from my babyhood, I was told that GABA-A phasic inhibition is crucial to any oscillation. And basically what happens here is that we killed phasic oscillation and nothing happened. So we were actually very shocked. We, of course, tried to quantify, maybe something comes out. And the only thing which came out that the oscillation slowed down with 1 hertz. I mean, so it's significant, of course, but compared to the fact that we were expecting a complete disruption of oscillatory activity, this is basically nothing. It just demonstrates that we have done something. So neither the number of cycles per spindle, the spindle legs, or even the occurrence of the spindle didn't change. So we were actually a little bit perplexed. But then we went on and teamed up with Anita Lüthi in Switzerland, and to figure out what happens with the burst iPSCs. So earlier investigators already showed that in case of the thalamic cells, if the inhibitory cells produce a single action potentials, basically you have a very tiny iPSC. However, if they produce a burst, then the amplitude of the iPSP is huge. Of course, many people thought before that this is basically because the individual action potential adds up, and that's why you have a much larger IPSP burst. But then we wanted to check what happens to these burst IPSCs, so we used a little bit different paradigm, and I think we found the key. So in contrast to the spontaneous IPSC and the single action potential evoked IPSCs, the burst IPSPs basically survived the elimination of synaptic receptors. Clearly, the amplitude decreased, but at least almost 50% of the charge has remained. And of course, how is it possible? You can't avoid the conclusion that extrasynaptic receptors basically got phasically activated and contribute to these burst IPSCs. These are just the quantification. It slows down, of course, and the amplitude is decreased, but the main point is that almost half of the charge transfer you still have basically mediated by the extrasynaptic receptor. So how we can explain it? That basically, beside the two well-known action of GABA, in this case, you have to add a third type of inhibition that during a very special case when the NRT cells burst, most probably these bursts induce a different release mechanism of GABA, and so much GABA is released that they physically able to overflow the synapse and activate alpha-4 and delta containing extrasynaptic receptors. And of course, this mechanism is utilized apparently for TC cells, and this mechanism is important for them to generate the rebound burst, which would start the next cycle of the oscillation. So how can we put this together? So it's clear that during burst IPSPs, GABA overflows and activates extrasynaptic receptors as well. It's also clear that this activation of extrasynaptic receptors alone is sufficient to maintain the excitation inhibition rebound, since we didn't see much changing in the spindle activity. But it's, as a consequence, what we think is basically going on, that under natural condition, when the spike per burst start to decrease, it reaches a critical level, most probably, and this is when the burst IPSCs switch to single IPSCs. And as I showed you, this will be a huge drop in the inhibitory conductance, and as a consequence, there will be a critical point when TC cell will not receive sufficient amount of inhibition to produce a rebound burst, and this is the time when the spindle is terminated. So the first conclusion, or the conclusion of the first part of the talk, is the proximate cause of spindle disruption is basically the switch of GABA release from the burst mode, where you activate extrasynaptic receptors as well, to a single mode where you activate only synaptic. Of course, the next question is that what determines this activity? And this is when we start to use optogenetics to figure out the mechanism. So the question is that why do you have this well-defined change in the activity of NRT cells? Basically, you can have three possibilities. Either you have an extrinsic input which somehow changes NRT activity during the spindle, and it will tell when the spindle should stop. Or the intrinsic activity of, or excitability of the NRT cells changes during the spindle, and this sets basically these changes. And finally, it's also possible that the entire thalamocortical network changes in a systematic fashion as the spindle progresses. So how can you differentiate between the two? So what we first did, basically we examined the first cycle and determined what is the probability that an individual cell fire in the first cycle. And we examined what is the relationship between firing in the first cycle and the duration of the spindles. And this is what we got. I think it's quite shocking. In case of the TC cells, you don't see much correlation. But in case of the NRT cells, you see an extremely strong reverse correlation, meaning that if you have lots of NRT cells, so lots of NRT cells participating in the first cycle, then almost all the time you will have short spindles. In contrast, if you have very few NRT cells participating, then almost all the case you have very long spindles. Of course, this comes to an almost philosophical question, because apparently this suggests that if there is such a strong correlation between initial firing activity and the duration of a transient event, then it comes that already when the whole thing starts, the entire system knows how long it will, for how long it will last. So basically, somehow the initial firing pattern already codes for how long the spindle will last. As a consequence, we can rule out the number one hypothesis. So somehow NRT should be involved in determining the duration of the spindle. So we have only the remaining two hypotheses, and I will go fast on these two, but just indicate if it's not clear. But we will use an optogenetic approach here, and as you will see, we will evoke spindles, both using VGUT animal and the PV animals, and we will activate the somata or the axons, and the results will be the same. And of course, the main idea is that we can vary light intensity, and via this we can manipulate the amount of NRT cells recruited. If you give more light, you will have larger population of cells recruited, and if you have few lights, there will be smaller population. And of course, we will measure the duration of the sleep spindles. First, what controls we should make if we use any optogenetic manipulations. In this case, we don't use repetitive pulses, and I think it's very important whether you can induce any spontaneous activity by a single laser pulse, and this is what we do. And as you can see, we apply a single laser pulse. These are different sweeps coming after each other in time, and as you can see here, with a single laser pulse, you can induce spindle quite reliably. However, not always. So as you see, and as you could hear before, the state of the cortical activity changes tremendously. So you can have desynchronized states, slow oscillation, and in between you can have these spindles in the light sleep. And apparently, if you give the laser pulse using desynchronized state or during deep sleep, you are not able to evoke spindles. And of course, this is a very strong indication that we are sort of inducing physiological spindles and not some artificial oscillation. But to control more, we basically compared laser-evoked spindles, the duration of laser-evoked spindles, and spontaneous spindles. And as you can see, they are very nicely matched, the distribution of the spindles. We couldn't, or we could very rarely evoke very, very long spindles, but all the other regions were okay.
So if you eliminate the single ones, when you get rid of gamma 2, why doesn't that have an effect on
yeah, but in this case, the gamma 2 is intact.
No, I understand.
Yeah.
You didn't eliminate the spindles when you got rid of gamma 2. It surprised you. You just would have created a world. When you previously started,
Yeah.
you got rid of gamma 2.
Exactly.
You said, oh, the oscillations are still there. I'm surprised.
Yeah.
Now you're saying that you need the single ones in order to augment the first ones to get the same oscillations that gamma 2 can do with them.
So wait a second. In the first part, indeed, we removed gamma 2. Oscillation remained intact. This is because the extrasynaptic, physically activated extrasynaptic receptors provided sufficient inhibition to promote the oscillation and to start the next phase of the oscillation. That was the first part. Okay, so, and, yeah. Okay, okay. Yeah, okay. And in these parts, we are inducing oscillations. And we vary the laser, laser intensity, and to check for it, whether it's really affects the system. As you see, as we increase the intensity of the pulse, the probability of inducing spindles are increasing, which is said that indeed light intensity affects spindle generation. You can also change the pulse duration. So after these controls, we can start the experiments, but I'll tell you the two hypotheses. So if there is a direct causal relationship between the initial NRT activity, you should see the following. If you use low laser light, you should evoke long spindles, right? Because low laser, because when you had so if you use low laser light and you are up here, then you will induce long spindles. And if you have high laser lights, you will induce short spindles, since high laser light is supposed to activate a lot of NRT cells. So that's why, if this correlation is true, then you will get only short spindles. The alternative scenario is that the entire system is oscillate between short spindle and long spindle epochs. In this case, if you are in a short spindle epochs, no matter what light you use, you will get short spindles. And if you are in the long spindle range, no matter what you use, you get long spindles. And these are the results. So in this case, the animal is sleeping naturally, and you activate continuously. You can see that sometimes laser activation doesn't work, and this is where you have deep sleep. But other times, the colored codes indicates that you use 5 different laser strengths. And I just show you here a weak laser light, which is supposed to induce a long spindle, but actually it induces a short spindle. And I also show you a strong laser light, which is, instead of inducing short spindle, actually induces a long spindle. So apparently, our hypothesis 2 is valid. So there is no correlation between the laser intensity and the spindle duration. And this is shown here in a whisker plot. So no matter what stimulation lengths you use, the duration of spindles are the same. So the overall conclusion, it's not the NRT cells alone which determines the duration of the spindles, but apparently the entire network is oscillation, and the laser light here simply kicks in spindle activity, and this is how we generate spindle. So the conclusion, so indeed, the duration of the spindles is determined at the onset by the actual state of the network. And of course, why is this important? As I told you, the duration of spindles is quite dynamic. It changes a lot. And when I learn something and I need longer spindles, it could be either controlled by the activity of the NRT cells, but apparently this is not the case. So after learning, apparently the entire thalamocortical system enters in a different spindling regime to be able to produce longer spindles. And with this, I think I have a couple of more slides where we compare conventional techniques, pharmocogenetics versus KO. But I think I may leave this and let time for you for questions.
I was wondering whether you were thinking of using delta knockout just to test directly whether the extrasynaptic receptors are the ones responsible for the spindles. And that would also be nice, because if you could get rid of the spindles, then you could test like some behavioral consequences.
Fortunately, it has been done by another group, and with exactly the same results. The problem is that they didn't check the spindles, but indeed, if you knock out delta, then you will basically miss the other parts of the burst IPSP. So it's the two study basically matches each other. So indeed, it demonstrates that the extrasynaptic receptors can physically activate it during an RT burst. But yeah, the expectation is that the spindles remain there as well. So but they didn't do it.
You think that the synaptic then would be enough to have the spindle?
I think the situation is that here the system somehow it uses a system when either mechanism alone would be sufficient to maintain the oscillation. So it's super sure in a way.
Two very brief questions.
Okay. Since the probability of the NRT neurons on the first cycle is correlated with the length with the spindle, did you check the peristimulus timing histogram of the spikes to see whether the neurons are really responding?
Peristimulus time histogram compared to the peak of the oscillation, or where?
When you stimulate with light to check what the raster plot triggered as the light.
So basically, I just try to recollect. So it's since we are always activating inhibitory cells, basically we can see a very brief activation of NRT cells, and then we have a long silence, which lasts for maybe 150 milliseconds, and then the oscillation starts. So it's not immediate. So yeah, we launch an inhibition and a rebound.
Very nice work. In addition to the duration, people also, well, there's evidence that the amplitude of spindles also changes after learning. So yeah, sure. I was wondering if you had any, what your perspective was on how the amplitude might also be changed.
You have the following problem in the amplitudes. Of course, in almost all human work, you have to record from scalps. I may not emphasize enough, but all these recordings are done in the thalamus, where the spindles are generated. So in case of the humans, you have a major problem of spindle detection, and different labs solve it in a different way. So the problem with the human data is that there are still no consensus how and where and what would the spindle, because nobody tells it, but there is a continuum between spindle and no spindle if you detect in the contrary. So you always have to threshold it, and that's why in this case we didn't pay much attention to the cortical spindles.
Okay, thank you very much.